Translation is a process that is preceded by transcription. Transcription is when we have mRNA being copied out from DNA, and now this mRNA, after it's been spliced with the help of spliceosomes, and it's now being transported to the cytoplasm where it can be translated. And notice that we're talking about eukaryotic translation. I recommend that you watch the previous video on prokaryotic translation, which will help you understand the differences between the two. And it will also help you understand that these two are very similar processes and they have the same end goal, obviously. So without further ado, let's talk about translation as an overall perspective first of all. So it has 3 steps, as you're probably aware of now. So first step is initiation. Initiation really is the process when we have different enzymes and these ribosomal complexes, the large and the small subunit, they come together and they realize that, hey, look, this is an mRNA that needs to be translated. This is the process called initiation, the recognition of the codons on the mRNA, which will help the entire enzymes and everything that's needed for this process to happen come together, and they all cooperate with one another. And we're going to see how that works. And we have elongation. Elongation is a process when we have the amino acid polypeptide chain. It's just going to be growing off, and that's the goal of elongation, just to help or just to read the open reading frame on the mRNA and grow the polypeptide through peptide bonds between the amino acids. And then we have termination. Termination is when we see the mRNAs or the ribosome will recognize that, hey, look, that's a stop codon on the mRNA, which signifies that the open reading frame is read, and now it's time to dissociate because the polypeptide is made. So now let's get into the detailed understanding of how initiation works. So initiation really begins in the small ribosome, ribosomal subunit. And what happens is we have an initiation complex. So first of all, we have a pre-initiation complex, the 43 pre-initiation complex formed. And this is composed of the 40S small RNAs, and it also has the methionine tRNA on it. So notice how we have, and this is not shown over here, but notice how we have methionine and we have formal methionine on prokaryotes, and that's one of the big differences between the two. And we have a lot of initiation factors that are doing different things, and we will talk about that in a second. I'm going to talk about some of them in a second. So the E over here just signifies eukaryotic, and this is I stands for initiation and F stands for factor. So eukaryotic initiation factor. So the next thing that happens is we have the 5' cap of the mRNA being recognized, and this will then be attached to the initiation complex of the proteins. And so once all the necessary pre-initiation proteins are found, so once everything is found and everything is together and the 5' end of the cap is attached to the mRNA, the next thing that happens is we have the ribosome scanning for the start codon. And once the start codon is reached, we have something called we have the entire initiation process completed and successful. And before we talk about elongation, let's get on to the proteins that are involved in the regulation and the process of initiation. So first of all, let's talk about the elongation. As an example, let's talk about the elongation factor 3. The eukaryotic initiation factor 3. So we talked about initiation factors. There are a lot of initiation factors. And this is one example to just show you that these initiation factors play essential roles. And the role for the initiation factor 3 is that it avoids the premature attachment to the large subunit. So small subunit needs all of its enzymes and all the initiation factors and everything to come together. It's a very complicated complex like we talked about in the ribosome video. If you haven't seen it, I recommend you go find it. So if all these proteins have to come together, or these enzymes have to come together, and before these enzymes come together, we can't have attachment to the large subunit, and the initiation factor 3 will make sure that doesn't happen. Here are some other 3 initiation factors we're going to talk about, and these 3 initiation factors are the limiting. So if you remember from chemistry class, we talked about rates, and we said that there's a limiting factor. These 3 enzymes these initiation factors are the limiting factor in how fast initiation can happen. So the eukaryotic initiation factor 4A, it unbinds the double-stranded RNA. So you might be confused, why do we have double-stranded RNA? And let's quickly think about this real fast. So some viruses have double-stranded RNAs, and think about it yourself. When we have RNAs transported from the cytoplasm to the from the nucleus to the cytoplasm, they're free. They can't be just by themselves. Think about it. Because imagine if you had a single-stranded DNA, it would be interacting with other things. And during replication, we have something called single-stranded binding proteins that will come and bind to the unwinded double-strand DNA, so it doesn't interact with anything else, and that's why we can have double-stranded RNA. So the initiation factor 4A will unbind the double-stranded RNA. And then we have the initiation factor 4E, which will the 5 prime, which will attach the 5 prime the 5 prime cap. It will attach it to the ribosome. And we have the initiation factor 4G, which is a scaffold protein. And the scaffold protein really just is a protein that is involved in the communication between cells. And we're going to talk more about scaffold proteins in future videos. It's a very important concept to understand later on. So the next step is elongation, and notice how we don't have I just want to highlight that again. We don't have the tRNA shown over here, but I just want to highlight the fact that the first amino acid of eukaryotic translation is the methionine amino acid, compared to the formal methionine, which is a modified methionine, which is used in prokaryotes. So please just watch the prokaryotic video and you'll understand what's happening over here. So we have elongation, which similarly has a lot of enzymes or proteins involved in its elongation. So we have something called the elongation factors, which will regulate translation. And we're going to talk about 2 examples of elongation factors. The first one is called the elongation factor 1. So the elongation factor 1 will aid in the attachment of the tRNA to the A site. So the tRNAs that are coming to the A site, they have to be attached through a bond, and we talked about this in the prokaryotic video. This requires energy, and because of that, there's a hydrolysis of GDP, and that will give it the energy required to attach to it. And and we talked about this in the prokaryotic video, that the enzyme involved in it has to be resetted so that the GDP is removed from that. So this is what the EF1, elongation factor 1, does as well. It removes the GDP and resets so that we have more GDP that can be used in the process of attaching to the A site. And we have elongation factor 2, which is just the elongation factor which is involved in just the translocation of the tRNAs, and that's pretty simple. And the entire concept of elongation, we've talked about it before, is to just elongate the entire amino acid, or the polypeptide chain. And this is what elongation does essentially. And then we're gonna have these, and we're gonna have elongation until the open reading frame is here, and it's gonna be looking forward to the termination sequence, which will indicate that this entire mRNA is ready and it's time to fall off. So here's a question that I want to ask you guys. Nascent polypeptides formed from polyribosomes in eukaryotic cells are all the same. This question is asking if we have a single mRNA that, of course, has been spliced, and it's a mature mRNA in the cytoplasm. If it's read by nascent or if it's read by polyribosomes, by a lot of ribosomes, and nascent just means that it's a new polypeptide. So all these different polypeptides, or ribosomes that are reading this mRNA, are they going to produce the exact same polypeptide? And the answer to this question is yes, because eukaryotic mRNA is monocistronic. And remember that we talked about polycistronic genes on prokaryotics compared to eukaryotes, which are only monocistronic. And recently we have found out that eukaryotes also have polycistronic. Sometimes, some of them, they have the ability to express polycistronic mRNA, but for the purpose of your biology class, and definitely for the purpose of the MCAT, we just want to think or just want to memorize that or really understand. Don't be memorizing this stuff. Yes, you have to memorize some vocab over here, but if you understand the entire process, then it should click in your head. So eukaryotic mRNA is monocistronic. So let's just understand that. So we have termination, and we talked about termination is when the stop codon is read by the ribosome. So the ribosome recognizes these stop codons, and it's gonna cause the entire ribosomal unit to dissociate, and the mRNA to be dissociated as well. And we have the polypeptide successfully completed, and it's ready to be. So we have something called post-translation modifications, and we'll talk about that later. And these post-translation modifications will allow so we also have these. So the polypeptide that's now produced is not mature. It has to be further it has to be further processed to be mature. And we're going to talk about that in future videos. We're going to use insulin is a great example, because when we, for example, have insulin produced inside the whole process of translation, the initial polypeptide for an insulin is not functional. It has to go through modifications before it becomes functional. So this is something interesting, and we'll talk about it later on. So getting back to termination. So termination is when we have the stop codon. Obviously, we talked about it before. So these are two termination factors we will talk about, and hopefully you can memorize it. So, or not, if not memorize it, just know that that we have termination factors. So the termination factor 1 will recognize all stop codons compared to the prokaryote. In prokaryotes we have different termination factors which will recognize different stop codons, but in eukaryotes we have the termination factor 1 which will recognize all stop codons, and that's very interesting. And we have termination factor 2, which is a GPDase, and it helps the TF1, which is termination factor 1, remove the polypeptide. So GPDase just means that it's causing the hydrolysis of ATP, and it's going to help the removal of the polypeptide. So let's talk about, before we end this video, we want to go through some questions real fast. So the first question is, which of the following statements are true? In eukaryotic cells, the following can happen simultaneously. And remember that eukaryotic cells have internal organelles. So keeping that in mind, let's answer this question. Translation and transcription. Now think about it. Why is this not possible? This can't happen simultaneously because when we have transcription, first of all, the compartmentation is different. We don't have the ribosome available inside the nucleus. And second, the mRNA is not mature. It's a heteronucleus mRNA, or the hnRNA, which has to be spliced through, or the introns have to be removed. And because of that, we can't have translation happening at the same time. Splicing of heteronucleus, I don't know why this is HC, is supposed to be HN, excuse me. Whoops. hnRNA. So splicing, splicing of hnRNA and translation. Do you think this is possible? This is not possible because, again, the location is different. We can't have splicing inside the nucleus, and we can't have ribosomes inside the nucleus. So this doesn't make any sense. These both don't make any sense. Transcription and splicing. This is possible because, think about it. If we have transcription and splicing happening inside the nucleus, so they can happen simultaneously. So this is the right answer. So hopefully you understand everything you need to know about eukaryotic translation, and it's really very similar. In the next video, we're going to compare eukaryotic translation with prokaryotic translation, not eukaryotic translation, excuse me.